Good afternoon, everybody. Thank you for coming. Welcome to the Stanford Cancer Institute Breakthrough in Cancer Seminar. My name is Max Dean. I'm a professor of radiation oncology. And today we have the privilege of hosting Dr. Simon Powell, chair of the Department of Radiation Oncology at Memorial Sloan Kettering. Our Breakthroughs in Cancer Seminar series is one of the highest profile seminar series for translational and clinical scientists in oncology. And we bring national experts to Stanford's campus each month to discuss innovations across the spectrum of cancer research. Dr. Simon Powell is the Enid A. Haupt Professor and Chair in the Department of Radiation Oncology at Memorial Sloan Kettering Cancer Center, also member of the Molecular Biology Program of the Sloan Kettering Institute, and Professor of Molecular Biology at the Weill Cornell Graduate School of Medical Sciences. Dr. Powell has many, many honors. Among those that he's most proud of include being an Astro Fellow as well as Astro Gold Medalist. Dr. Powell's research has highlighted cancer-specific defects in DNA repair and the DNA damage response, which have provided opportunities for therapeutic exploitation via synthetic lethality. In particular, he's had a long-standing interest in homologous recombination in human cells, which led him to do deep investigations of the role of the breast cancer susceptibility genes BRCA1 and BRCA2. Along the way, he developed functional and genomic assays of homologous recombination to be able to identify cancers that have this defects in this type of repair, and I suspect we'll be hearing about some of that today. And he also identified that human RAD52 is synthetically lethal in cancer cells that lack functioning BRCA1, BRCA2 pathway, and this has led to novel therapeutic strategies targeting RAD52 in that context. With that introduction, it's my pleasure to invite Dr. Powell up, and we look forward to his talk. Thanks.
Okay, thanks very much for that introduction, Max, and it's great to come back to Stanford. I get to come here almost once a year because I'm on the Stanford Cancer Institute Advisory Board, which is something that's I have great pleasure doing. So yes, homologous recombination deficiency is a cancer phenotype that I want to convince you is more prevalent than you first appreciated. And there are now many ways to establish that diagnosis. And over the next window of time, we want to do even more to try to establish it as an important, not just phenotype in breast and ovarian cancer, but in a broader array of cancers. This is some data that was just published in Nature Genetics, largely in relation to ApoBec, which is not highlighted. But this is just taking 4,000 breast cancer patients who've had MSK impact, which is just the 500 gene testing. Is there a laser here? Should have checked. That's the laser, yeah. Oh, can't see it. Anyway. The 500 gene sequencing, and there is enough sequencing information from that MSK impact to recognize key elements of either ApoBec signatures or homologous recombination signatures in terms of both the single nucleotide variants as well as the indels. And you can see, whereas you would think that you would see HRD, which is almost 40, possibly even higher in metastatic disease, you wouldn't have expected to see it in HER2 amplified, which it's clearly prevalent, but even in ER positive, so the common ER positive, HER2 negative, still has 10% that may well be HR deficient. Well, numerically, that's 300. It's as many as the triple negatives. So the point I want to make is that this is a potentially prevalent condition. And if it's prevalent in common cancers, it may have a much broader footprint than we've seen. And we've started to look into this. This is looking again at MSK impacts HRD score shown here. And then this is the myriad score. I think some of you, particularly in the clinic, may be familiar with myriad assay that's largely based on various types of large-scale loss of heterozygosity. And if you look at this score versus the HRD that you can extract from sequencing data, like I showed you on the previous slide, you can see that obviously the vast majority correlate, which is good, but you can see that there's also some separation. And for a biological marker, you don't want this degree of, if you like, false positives or even false negatives. And if you look at the concordant ones, the ones in which, if you like, it's real in both assays, it predicts the response to PARP inhibitors, whereas ones down here that will be high in the myriad score will show a discordant behavior or will be disappointing. So we need to improve our diagnostic certainty, and we're trying to get to that by taking the now 140,000 tumors that have been sequenced on MSK Impact, looking at the prevalence of HRD and validating as many of them as necessary with whole genome sequencing. And in the whole genome sequencing, we'll also get at what additional factors what additional information whole genome sequencing adds to what you can get from target gene sequencing. So, and again, this just shows that in whether it's breast, ovary, pancreatic, or prostate, you can see that the germline biallelic, which are the current indication for PARP inhibitors and are used widely, but the somatic biallelic, in other words, no germline inheritance, but you've got two hits, they behave basically just like the germlines. But it's certainly in breast and ovary, that's not currently an indication for PARP inhibitor treatment. So again, you can see we're arguing to increase the footprint of indications for treatment in homologous recombination cancers. And then, obviously, this is a slide from Serena Nick-Zainal's paper for what she called HR Detect. This is a better algorithm than Myriad's HRD because it integrates the single nucleotide variants, the indels, and the structural variants into a single score, and essentially shows that if you have an HRD score of greater than 0.7, I won't go into the details of the scoring method, you get the same characteristic. You'll find some known germline, but you'll also have some somatic BRCA1 and 2 mutations or methylations included in that group. So again, giving the same message. So there are ways to make this diagnosis, and that's something that we're looking to do. This are two figures abstracted from this paper we published 2 years ago now, where we used long-read DNA sequencing and identified some rarer features of BRCA-associated cancers, but if you like, they're rare, but more pathognomonic. And again, one of the things we want to do in future studies is ask whether these rare pathognomonic findings are stronger predictors, or does the sort of common data that you can get from the target gene sequencing. And what we showed here is reciprocal pairs. This is basically where a chromosome break has a reciprocal paired event with a second locus, such that you either get reciprocal duplications. So here you can see a short duplication at the both loci, or in this case, reciprocal duplications at the paired loci. And these are triggered by presumably one initiating event. It goes against the common chromosomal dogma that you need two breaks to see a chromosomal translocation, because this has to be initiated by one event that triggers the aberrant junction, and then the reciprocal event is a product of the initiating event. And you can see that in BRCA1 you get these reciprocal duplications, in BRCA2 you get these reciprocal deletions, but you get a mixture in both of them. And these are less common, but absolutely, look at the p-values here, they're completely off the chart in terms of significance. And then a second feature that will be relevant later in the talk, which is BRCA2 genomes specifically have evidence of annealing deletions which have a degree of homology at the junctions, but they weren't previously recognized as deletions with homology because they have, on average, you can see about 50 to 60 base pairs of homology, whether you use long read or whole genome sequencing, but there's about 10% of mismatches in the annealing. So annealing is promoted even with mismatches, and it produces, again, a feature of BRCA2 tumors that, again, has a p-value of 10 to the minus 26 or something extreme. I can't even read it. Sorry about that. It's your projection system, I think. Anyway, it's in the paper, so you can read it if you want to. But it's highly significant. And again, we wouldn't have seen this feature unless we'd done the long read sequencing to walk across the junctions. And again, this feature is not as common, but it's very diagnostic. And this one is diagnostic of BRCA2 only. BRCA1 has a resection deficiency, so you wouldn't see those single-strand tail annealing. And then this is simply to show that our new features, which are shown in red, add to the features that you have with HR detect that I showed previously. And with those combined features, you make ROC curves that are very respectable diagnostically to distinguish BRCA1/2 deficient from the rest, or even to distinguish BRCA1 versus BRCA2. Some of the other algorithms done that, such as CHORD, but again, these features allow both BRCA1/2 from normal and BRCA2 versus BRCA1 because of those new characteristics. And you can see this little algorithm shows the relative contribution of the new features to that distinction. So obviously one of the questions we want to go on to answer now is, can we prove that HRD is clinically significant? Now, we want to use the MSK impact data to do that on a much larger scale. Invite me in a few years and we'll have the answer to that. But in the meantime, we did this study, 15032, so you can see it was a while ago, where we just used platinum and radiation for terrible triple-negative breast cancers, metastatic or locally recurrent, multiply treated triple-negative breast cancers. Now, what made this data set interesting is that it closed eventually with 49 patients. I'm sorry, I should update this slide. If you go to the San Antonio Breast Conference, I'm going to try and get all the data in order for that meeting. But anyway, apologies for the fact that the data is not fully analyzed, but the point I want to make from it is fairly clear. In this data, we took these recurrent triple-negative breast cancers, diagnosed homologous recombination deficiency by a functional assay, which is the ex vivo RAD51 assay. This has been published multiple times by me and other groups, and it's a test of live cell function. I won't go over the details for the sake of time, but we can categorize people with this assay. And when you do that, you can see that if you're HR deficient, your response, even in these heavily treated patients, this is imaging response, is 78% where you give platinum and radiation. And if you're HR proficient, it's 21%. So the difference between the two is huge. And this is just old-fashioned platinum and radiation, which actually is very good at exploiting the HR deficiency phenotype. The combination does better than either agent alone, something we actually just published recently for those who want to look it up. But if you just look at the response, if you have metastatic disease outside of the radiation field, you can see that the response to platinum only is a lot weaker, which is sort of goes with the clinical experience. So that's a segue to say making the diagnosis important. Yes, it does discriminate, particularly if you're using homologous recombination directed therapies. And obviously, with the advent of PARP inhibitors and others coming, Pol theta inhibitors, I think the opportunity to do that is going to get larger and larger. So now I want to focus on some of the background scientific concepts. Obviously, for those of you who don't eat, sleep, and breathe DNA repair, this is your quick refresher course. This is a symbolic double-strand break, just shown by simple strands. In actively proliferating cells, you get rapid processing of the ends, 5' to 3' end resection, revealing 3' tails. Non-homologous end joining, particularly in non-proliferating cells, don't undergo 5' to 3' end resection and will repair these as blunt or near blunt ends with small deletions only. If you have homologous recombination, you use the sister chromatid or another homologous sequence to repair the damage and will seal this with high fidelity. Obviously, if you lack the BRCA1/2 pathway of homologous recombination, it goes all the way down through paralogs. You get shunted into these annealing pathways of alternative end joining, MMEJ, and I also today want to include single-stranded annealing in that spectrum of what I call these large downstream deletions. And some of the synthetic lethalities are obviously linked to this salvage pathway. Should point out that none of the NHEJ machinery is synthetically lethal with HR. So it's not that they play no role, but knocking out NHEJ does not produce synthetic lethality. So there's a lot of opportunity for synthetic lethality in these cancers. The so-called backup repair pathways can be listed here. These days, there could be more than this, but the list is already plenty long enough. Base excision repair is still relevant, especially in replication-associated lesions, and of course was the initial proposed mechanism of how PARP inhibitors were synthetically lethal with BRCA, although PARP inhibitors almost certainly has a broader brushstroke and is involved in many more repair pathways than just base excision repair. I'm going to focus in a minute on this spectrum of what I've called ALT-J to cover MMEJ and SSA. There's been a lot written about RNA-DNA hybrids and their ability to negotiate G4 hybrids. For those of you interested in this, apologies. I'm not going to talk about them today. I don't know who there is out there. But anyway, I will talk also about some new data that I don't think any of you have seen on daughter strand gaps and as a precursor to the translesion synthesis polymerases. We've known in synthetic lethality screens that the TLS polymerases come up. And Max also introduced the role of RAD52. And just one of the things we want to get back at is just exactly how RAD52 is synthetic lethal with this collection of tumors, because there's a number of potential candidates. So that's the outline of the remainder of the talk. There's also another area that's of interest, some of these mitotic missegregation, CYP2A and TOP-BP1 also come up. And again, so the landscape of opportunities for targeting these tumors is really quite high. So here's the first new data that was a big surprise to us. Here is Maria Jason's standard assay of direct repeat GFP reporter. But instead of looking at homologous recombination, it's tuned to looking at single-stranded annealing by virtue of having a 3' deletion in this copy of the gene and a 5' deletion in this copy of the gene, such that if you make a break here, you will anneal between these homologous sequences and reproduce a full length. This is annealing repair, not homologous recombination. In other words, it's specifically designed to not undergo homologous recombination. And if you then screen, we were interested in what was contributing to the later stages of annealing. So once you've got resected ends, anneal through whatever amount of homology, you either have to clip flaps. We know that that's probably ERCC 1 XPF, just like it's RAD110 in yeast. And then there's these gap-filling polymerases, which we wanted to understand. And this was the introduction. We looked at a whole bunch of potential gap-filling polymerases. And to our big surprise, one of the best hits that came up was PolQ or Pol theta. Now, we did not expect PolQ to have this role. You can see that some of these other interesting polymerases actually have, if you like, the opposite effect. I won't go into that today, but these are quite interesting observations on their own. But today, I want to focus on this unexpected effect of PolQ. If you then deplete BRCA2, the number of annealing events goes up hugely, and you can see it's clearly PolQ dependent. So this is a massively unappreciated effect of PolQ, that it's working on single-stranded annealing, not just MMEJ. And the one I skipped over is that obviously the inhibitors were doing the same thing. Now, it's been long known that RAD52, my favorite protein, is also involved in single-stranded annealing. And both of these can contribute to this annealing defect. Here we've slightly changed the assay using a CRISPR double-strand break. And we analyze sequences that emerge from the repair of these double-strand breaks and put them into different categories of 5 to 25 base pair deletions, larger deletions. And what I hope you can see is that when you deplete BRCA2, you get an increase in these larger deletions, just like we would expect. And you can see that they're both PolQ and RAD52 dependent. And again, they're also PolQ inhibitor dependent. So again, not just this rather large homology segment is PolQ dependent. If you like, there's a spectrum as you go from microhomology to intermediate homology to larger homology. They're all PolQ dependent. And that is really a much different view of what we thought PolQ was doing. So that was a very novel observation that we certainly wanted to pursue in more detail. Here is just what sequencing data of these CRISPR repaired junctions, where you can see in the non-target or control, you get mostly NHEJ events and a few of these homology-based annealing events. Here's the homology. This is the deletion size. Deletion size is getting greater. And this is a zoom in on just this area. What you can see with BRCA2 is that obviously this area expands. We'd expect that. And again, what you can see is that these deletions, these larger deletions, are expanded in the BRCA2 deficient compared to this. But if you then deplete PolQ or RAD52, they all revert back to the previous, the phenotype. So these are clearly both PolQ and RAD52 dependent. Now, next we ask the question of what was driving this. PolQ is this interesting protein that has both a helicase domain and a polymerase domain, as well as this central domain, which of course has one of these beautiful intrinsically disordered domains, which always adds intrigue, but we don't actually know what it does. As you'd expect, some people have reported it existing in aggregates, but again, I don't have a comment on that. We don't know just exactly how important the central domain is at this point. So for the purposes of the point I do want to make is that here is the same assay looking at these indels, and here is the polymerase mutant, no effect. Here is the helicase mutant, all the effect, and the double is the same. And this is the double-strand break assay. This is the Maria-Jason single-stranded annealing assay. And I think you can see that the observation is the same, whichever assay. So this, number one, reinforces that you need PolQ for these larger annealing deletions. And even though I started this by looking for the polymerase, and we'll get, that's another subject, it's actually the helicase activity of Pol theta that is driving this. And then the final part of this section is just to look at, now here's the double-strand break assay again. Here now we're looking at the flanking regions, and now instead of looking at pure homology driving the annealing event, we're now, just like I showed you in those BRCA2 genomes, we're looking at like small versions of homeology. In other words, you get little mismatches. So here's an example of like 12 base pairs with 2 base pairs of mismatch. And ask yourself the question, do these come out of this assay? And the answer, of course, is yes. If you are looking at, once you're looking at deletions larger than 25 base pairs, you can see that the homology-driven deletions are small, and these homologous events are actually much more frequent. And again, they're PolQ dependent. So that phenotype that I showed you in the BRCA2 genomes in a larger scale, actually you can see them all the way down to 10 to 12 base pairs of near homology. And it may, again, this could be used as part of the diagnosis of BRCA2 associated cancers and possibly even BRCA1. But okay, so does this occur in real cancers? Yes, otherwise probably I wouldn't be showing you. Here you're looking at 5 to 25, 25 to 200. You can see here we're looking at homology, and here we're looking at homeology. And you can see the BRCA effect is much more obvious when you look at homeology as opposed to the homology. The effect is still significant, but it's smaller. And again, if you use HR detect high-low, you get the same result. So this homologous deletional annealing is actually a common phenotype, and it behaves in the same way as other annealing repair events, and they're all PolQ, helicase, and Rad52 dependent. So again, in terms of thinking how you would potentially use these in a synthetic lethal strategy, you can see that you've got your double-strand break, you've got your resection, you've got your RPA coating. We don't know yet whether the Pol theta helicase is needed for, if you like, stripping of the RPA. That's certainly one role that's been thought for it. And again, you get either small or large segment annealing, and then you get gap filling. There may well be a role, like Dale Ramsden has shown, for Pol theta in initiating some aspect. But again, there are other enzymes that can take that over, such as Pol lambda And again, it shows that the Pol theta polymerase activity is not essential for producing those lesions. So the takeaway of that part of the talk is that Pol theta is doing more than we thought. And of course, as a target for homologous recombination deficient cancers, it may well be, it may well, if you like, improve its efficacy because it's essentially attacking the full spectrum of the backup repair pathways for double-strand breaks that was not previously appreciated. So now, in the next segment, I want to give this new story, another new story about daughter strand gaps. Like many investigators, ran a whole genome CRISPR screen comparing BRCA2 deficient with BRCA2 wild type. We've represented it in this plot where essentially non-essential genes are here, essential genes are here because they kill everything. And if you've got no specific effect on BRCA minor cells, you fall along this diagonal. The more you fall away from this diagonal towards the specific to BRCA2, the more it's a synthetic lethal hit. And what you can see here is not only POLQ that I've just talked about, and a bunch of other interesting proteins here, such as MAD2L2, which some of you will know as Rev7. But the part I want to focus on now is that you see RAD1, HUS1, RAD9, and again, also TOPBP1 may well be a cooperator in this result. But RAD9, RAD1, HUS1 forms a heterotrimeric ring complex, which is, if you like, analogous to pCNA. pCNA, as you know, is the universal clamp for the replicative polymerase and is loaded on the DNA by the RFC 1 to 5 complex. The 911 is a heterotrimeric ring. It's loaded by the RAD17, substituting for RFC1, but is also a clamp loaded. And here, biochemically, it is thought to load on the 5' end of a double-stranded, single-stranded interface. Whereas pCNA, of course, loads on the 3' end for recruiting the replicative polymerase. And so we asked the question, the fact that this came up high in our synthetic lethal screen, could we validate that? And the answer, of course, was yes. Here we did siRNA for RAD9, HUS1, RAD1. No effect in the wild-type background. These are DLD1 cells. I don't think the cell background because we've reproduced it in other cell backgrounds. And this is we use that cell because it had a well-characterized isogenic BRCA null partner, and you can see the significant reduction in cell survival. And then if you make a DLD RAD9 null and then deplete BRCA2, you see the reciprocal effect. So again, it validates. You may notice that HUS1 null is not on this list. That's because it's essential. So HUS1 clearly has a different, an extra role in addition to its role in the 911 complex. And here is just to show that RAD17 has the correct phenotype. Here you can see that non-significant effect in wild type and significant reduction in plating in the BRCA2 deficient. So all of the validations supported the screening observations. Single-stranded DNA gaps are highlighted in BRCA deficient cells. There's a whole series of publications from various groups, Sharon Cantor, Alberto Ciccia, Alessandro Vendini. So there's a lot of interest in these single-stranded gaps in BRCA deficient cells. So we then asked the question using the S1 nuclease assay, which is basically an assay of single-strandedness. S1 nuclease cuts single-stranded regions in the genome. If you've got a large amount of single-stranded gaps, the average length of your IDU pulsed fibers will go down, and you can see the effect when you have the double deficient cells. So although there's a small effect of BRCA2, the effect in the double deficient cells is really, standout. So you can see here and here a highly significant result. So supported, or at least was consistent with gaps being part of the story. So again, Primpol has been highlighted as one of the mechanisms for restarting these gaps. And again, here what you're looking at is when the replication fork comes along, it can be the lesion that was produced here can be reprimed by Primpol beyond the lesion. And what you can see here, and you are in the audience. You were right. So you can see that in the wild type cell, it doesn't change. So it is BRCA specific. Thank you. I did look at it beforehand. So whereas Primpol has little impact on this S1 nuclease assay, or if you like, an assay of gaps, again, this is the result I showed you previously, and then this is what happens if you deplete Primpol, showing that in the BRCA deficient lines, or in this case, the double deficient lines, you can see that the Primpol effect, that the Rad9 double effect is Primpol dependent. And you can see it here and here. So this supported the model that these gaps were being made and that the gaps were Primpol associated. So we then asked the question, are these gaps being repaired by post-replication repair? And in this assay, what you're doing is pulsing with IDU, unifilar labeling rather with IDU, and then doing a shorter pulse with CLDU. So anywhere there are gaps to be repaired, you see as a green dot on the labeled chromosome background. And when you do that, you can see that in the BRCA2 deficient cell, you get post-replication repair of those gaps because that's what you'd expect. The gaps are made and they are closed, and you can see increased synthesis. This result has been reported before by others. However, in the double mutant, not only do you see the effect of obviously the Rad9, the Rad9 relative to non-target is not significantly altered, but instead of being stimulated, it actually goes down. Somehow the translesion synthesis doesn't happen. It doesn't get recruited. And the same is true for Rad1. So we then wanted to measure the gap length, and we did that now by again, unifilar labeling with IDU, and then the DNA here is stained by yo-yo1. So anywhere the DNA is single-stranded, you can see it because the IDU antibody will see it, and therefore you can see the number of the lesions and you can see the length of them. And this is utilizing combing as opposed to replication fibers. And by doing that, you can plot the gap length. And what you can see is gaps get bigger with depleting BRCA2, but not a huge effect. But when you have the double effect, you can see it's much enlarged. So what the 911 complex appears to be doing is that the gaps are getting bigger, they're getting more numerous, and gap synthesis is inhibited. We then ask the question, because biochemically we know the 911 complex binds at the 5' end of a gap, we then ask the question, are the gaps being enlarged because the 911 is protecting the end? And the enzyme that enlarges or digests 5' ends is primarily XO1. It's the most potent 5' to 3' exonuclease. And what you can see here is that this is, again, just cell viability. This is if you like, the data I've already shown you, the loss of cell viability. When you deplete XO1, the viability is restored in both RAD9 and pretty much in RAD1 as well. So again, that supported the idea that what the 911 complex was doing to induce loss of cell viability was by protecting the 5' end from degradation. It wasn't MRE11 or DNA2, other potential candidates. Again, sorry that's small, but again, it's the control to show that those exonucleases had no contribution to restoring viability. And then we ask the question, does XO1 help restore the S1 nuclease defect? In other words, the gap defect, and the answer was yes. We asked the question, did it restore the gap length defect using that assay? And the answer was yes. And then finally, did it recover the gap synthesis assay? And the answer was yes. So essentially, the lethality is directly linked to this frequent gap lesion that's seen in homologous recombination deficient cells. Although this has all been done in a BRCA2 system, much of this may well be true in BRCA1, although the exception in BRCA1 is that BRCA1 appears to have an XO1 defect. So that's something we're looking into in more detail. So in summary, what I've said here is that the 911 complex is loaded on the 5' end, PCNA is loaded on the 3' end, but there's something about the presence of the 911 that helps the translesion synthesis be recruited to help close the gap from the 3' end. And that's something we're still looking at. We've got pretty good evidence that it's Pol Zeta dependent, although there may be some redundancy in that part of the pathway. But it's encouraged us to think that 911 might be a viable target. And in collaboration with Mike O'Donnell, we've already looked at the site of binding between RAD9 and RAD17 that you need for loading and have a high throughput assay that we can screen for compounds that perturb the 911 RAD17 interaction, which would be specific for this pathway. Oh, so I probably said most of that. So yes, it needs, somehow there's a connection to the gap, the TLS, the translesion synthesis machinery to help close the gap. It seems to be preventing XO1 degradation. Still looking at the role of top BP1. I didn't show much top BP1 data, but top BP1 does show a synthetic lethal phenotype, but it could be through interactions with other potential pathways. This is another big question we want to ask, which is, are the single-stranded gaps toxic by themselves, or do they need to be converted to double-strand breaks to be toxic? And we're looking at potential candidates like ApoBec, enzymes that would convert, or other enzymes that could convert stretches of single-stranded DNA and break them. The longer they are, the more likely they are to break. So I don't have any data on that yet, but we're certainly actively investigating it. And again, we're looking obviously how they play a role in repairing these BRCA2 specific lesions. Okay, then finally, just wanted to make a few comments, not a lot of data, but a few comments about what we know about RAD52. We know that one of the things that we've been trying to study for a long time has been a bit controversial in the field is I've always felt that RAD52 plays a role in RAD51 dependent HR as a backup RAD51 dependent pathway. The yeast people in the DNA repair field have always debated this because for whatever reason, human RAD52 won't biochemically perform that reaction, whereas yeast RAD52 will perform it very nicely. And we've still not quite solved that problem. But I keep reminding that this was the data I published 15 years ago, showing that, yes, if you deplete RAD52 in a wild-type cell, the RAD51 foci, RAD51 foci are not particularly affected. Sorry, it's not labeled as green is RAD51. But if you deplete BRCA2, obviously you vastly affect the RAD51 foci, but there are a few S-phase cells, and this is all published, S-phase cells that have some RAD51 foci, but if you then deplete RAD52, they all go away. So I've spent the last 15 years trying to prove that RAD52 does have a function in RAD51. This is the recombination assays that also supported. And these recombination assays have to use RAD51. They're clearly a RAD51 dependent activity. So recently, and I'll go quickly, I see I'm running out of time. This is an assay we've used before where we use a double ISC break that makes these incompatible ends and promotes resection. This is just to show that, whoops, what have I done? That's not good. This is just to show that the time course of developing DRGFP, if you use this type of system, occurs, takes about 17 to 30 hours before you start to see significant recombination, whereas the breaks are made, and again, this is just, the breaks are made, within about 7 hours. So what happens between 7 and 24 hours? So we developed this assay, which was a variation of an assay that Wolf Heyer had developed in yeast, where we wanted to not only study resection, which is a different subject, but we wanted to study these junctions that you could make between a broken resected end and a homologous template. And in this, I used a plasmid recombination substrate so that I could control the sequence. But what you can do in this assay is invade the homologous donor, cross-link it, and again, the details I'd be more than happy to go over. But again, when you get this junctional molecule made in that 7 to 24 hour window, you can cross-link it, cleave these ends here and here, and make a proximity ligation product that has a unique PCR product. So it's for those of you who didn't like the details of the assay, I apologize, but essentially the PCR product is only made when you have a junction molecule. That's the takeaway. And does it work? Well, obviously I wouldn't be showing you if it didn't work, but it did take quite a long time to make it work. And you can see these intermediates come up right where you would expect them to come up. In yeast, this happens within 3 hours, but in humans, this is in the 9 to 18 hour window where you see these recombination intermediates. These recombination intermediates are dependent on BRCA2 and RAD51, so we think it's, the real thing. And, oh yes, this is some new data I just threw in, which is the paralogs all affect this assay, just like BRCA2 and RAD51, with the standout exception of XRCC3, that although it has a recombination defect, has no defect in this capture. That's of interest to the people who really know this field, but it does appear that the RAD51 paralogs have two different complexes, one that is BCD2 and one that is XRCC3CD XRCC2. And so it supports the idea that there may be two paralog complexes working at different stages. And now back to RAD52, you can see that in this assay of capture, RAD52 is clearly having an effect. So again, I'm showing this to all the skeptics in the field that didn't think RAD52 could support RAD51 dependent function, because here it seems to clearly support that function. And I'm going to, I'll just skip that. I like that one, but I want to skip it so I don't run out of time. Sorry about skipping over data. So when it comes to RAD52, the question we're posing now is that we've shown a number of functions for RAD52. There's obviously its role in annealing, which is RAD51 independent. There's the role of RAD52, RAD51 interaction that has been mapped to this region of RAD52. That by mutating this region, we can separate the RAD51 dependent functions from the annealing functions that are associated with the N-terminal domain. And then recently, we published a paper showing that the C-terminal domain of RAD52 played a role in transcription replication collisions. That was published in Nature Communications, and I'm sorry I didn't have time to talk about that today. But again, that we could localize to this domain. And so what we're now working on is asking the question, is the lethality linked to annealing, to RAD51, or is it this role in transcription replication collisions? And by mutating this place, this and this place separately, we should be able to dissociate those different functions. Sorry, I'm skipping that one. That's just that's in the transcription replication collision paper. So that's really what I'm showing you. That we're really, I don't have that data yet, but we're going to really try to nail down which is the critical element of RAD52. Obviously, I mean, it could be it could be any or potentially all of the above. So what are my overall conclusions? I hope I've tried to convince you that HRD cancers are common and can be seen by using genome-based diagnostics. We're ready to expand HRD-directed therapies. I've shown you that annealing isn't just MMEJ and SSA, it's really a spectrum. And actually, they have a lot in common. The classic MMEJ assay is not RAD52 dependent. The little junctions don't bind RPA, and probably for that reason are not RAD52 dependent. But once you get above 25 base pairs, they all have similar dependencies. I've also shown you that gaps are a feature of HRD cells, and that the 911 complex appears to play a role in protecting the 5' ends. I didn't focus on this, but one-ended double-strand breaks are a potential reason why some aspect of RAD51 function may be required, whether it's through well, there's a number of potential components what might require RAD51 function there, but again, this is another important component to look at. And the replication fork collisions with transcription complexes has been shown to be a source of lethality. And again, I didn't show you much of that work today, but it's certainly all in that publication. So could you have future combination therapies for HRD cancers? Well, PARP inhibitors and POLQ inhibitors are already happening. They're in the clinic right now, so we'll see if that's a useful impact. could you do RAD52 and POLQ inhibitors? Well, I'm not sure yet if RAD52 inhibitors could be made. And obviously, I'm also hoping or optimistic that maybe this interaction could be harnessed. Here is a very quick summary of my RAD52 inhibitor development. Compound screen showed a lead hit. It had impact on SSA, which was a good readout of RAD52 function. 2 biosimilars, one has no effect on survival, one has a huge effect on survival. This one didn't affect SSA, this one did. When we take this one, compound 56, and treat wild-type tumors or BRCA2 deficient tumors, it does nothing to the wild-type tumors. It pretty much arrests the growth of the BRCA2 deficient tumors, and it does that without any significant weight loss. We took this compound to our Drug Discovery Institute. They said, that's a shitty compound. We'll redesign it for you. I said, what the hell are they doing in the library then? Because, what's the point in using them in a compound screen? But I think it's proof of principle that the system can be targeted, and this was screened against a sort of DNA annealing assay. So sorry, I've run a little long. Just want to acknowledge Helen Grimsley did the 911 work. Manisha did the POLQ SSA work with help from Erica Buchelmeyer. And there's the happy lab. Oh, yes, that looks like the same guy in the original photograph. And there's the group. You see, it's not that small. Anyway, the happy group on one occasion recently.
Yeah, great talk. So for your first part with the polymerase beta inhibitors, were you using Novobiocin or the Arctos inhibitors? Because I think they're targeting, like these Arctos ones, I thought they were targeting the polymerase part, right?
Yeah, I didn't get into the nitty-gritty on the different inhibitors. Novobiocin in our hands doesn't work very well. We've used, the Artios compounds that were available were polymerase inhibitors. They do have some effect. So the genetic result is much cleaner than the drug result. The genetic result is really clear, but most of it was done with, because I collaborate with Danielle, it was done with RP3467, which is the drug that's in the human trials, right? Which obviously we think is good. That's the broad we, not me particularly. No stocks in repair.
Yeah, beautiful talk. I'm just wondering, you didn't talk so much about between, impairing DNA repair and then the eventual apoptosis. So depending on how you go after repair, how different is the apoptotic, induction?
Well, I think most of these cells don't really die by apoptosis. I would say they mostly die by some form of, I don't like the phrase mitotic catastrophe, but they certainly go through a post-mitotic event. So these basically die from just genomic screw-up. And they're mostly post-mitotic. Then there's very little interphase apoptosis. They're all P53 mutant.
And in terms of prioritizing them in terms of therapeutic index and other things, what's your thought process on that?
Well, the the arguments in favor of POLQ or Pol theta are fairly strong. The expression is much higher in tumors relative to normal cells. They don't appear to produce too much background DNA damage in normal cells, which of course is not true for PARP inhibitors. PARP inhibitors produce a lot of nicks and other types of damage that you can see in any cell that's proliferating. RAD52 as a target is a great one if I could only find a drug, because if you treat a normal cell with a RAD52 knockdown or a or that compound that they told me was lousy, it really does very little. You can't detect DNA damage in that background, right? So I think the potential for avoiding background damage with Pol theta and Rad52 is probably much better than PARP inhibitors. And if PARP inhibitors start to develop second cancers, and there's been 2 reports of that now, it will bring they may become more in the center rather than an adjunct in the future.
A very nice talk. In the early part where you're identifying this new HRD signature, do you always find the mutations that explain that signature, or are there some cases in the tumors that you find the signature and you can't explain it in terms of the genetics of it?
There's a few that you can't explain. Methylation accounts for some of the non-genetic. Methylation is higher in ovarian than breast. May come up more, as the tumor evolves. I think the opportunity for methylation probably also increases. We did a whole screen of BRCA1 methylation in de novo breast cancers, and the de novo rate was not that high. So, there's a few that you can't characterize where, but if you look at the paper we published, what is it now, 6-7 years ago, you can explain probably 90% genetically. Genetically or by epistatic inactivation. So that type of genetic. The number that you can't explain is probably only about 5%.
And on the therapeutic side, what about the, have the embarrassment of riches of ways to target this deficiency? Any thoughts on which of those options is best for combining with something like radiotherapy or sensitized chemotherapy as a sensitizer that doesn't sensitize the normal cells, of course?
Well, there's a lot of experiments to do, right? I don't, whether the best way to treat HRD in the future is going to be sort of targeted, specific targeted therapies, mono or combination, or whether ultimately they're going to work better with radiation or certain types of chemotherapy. I mean, the cynics would call, PARP inhibitors weak chemotherapy, right? Because they produce a fair amount of damage. I don't think we know that yet. Obviously, if you go to the other extreme and you look at DNA PK inhibitors, they don't do anything unless you've got, radiation or something to act on. So it may be that they may do better if they have something to act on, and it could be, finessing a low dose with the inhibitor may well prove to be the best. But I honestly, I wish I knew the answer, but I know we can do the experiments.
Great. Okay, any other questions?
Anybody online?
Questions online?
Okay, one more.
One more question back here? Okay, yes, one more.
Colleen.
Really nice. I'm curious about the mechanism of resection by EXO. Do you think that 911 is sufficient to block this? Can you reconstitute this, or do you think it's recruiting something that's inactive?
Yeah, I don't know. I mean, obviously, I've only with the genetic data shows that the EXO1 double data is pretty good at reversing all of the phenotypes we've seen. But obviously, I don't answer. You're asking the more sort of operational structural question, which is what else needs to be there. And obviously, I mean, I was hand waving that if you keep your gaps to a certain size, there's obviously some connection between the 911 and the TLS recruitment. I mean, an obvious candidate is top BP1, right? Which we know gets recruited to the 911 complex. That's got a whole bunch of BRCT domains that could interact with a bunch of other proteins. So, and we know that plays a role, but I don't know there could be other players that are going to contribute to that. Don't have those answers yet. Do you have anything? Did you have a specific protein in mind?
No, I was just, it seems like you're implying that the 5' end influences the 3' end.
Yes. That is that exact. Well, I've talked to people like Mike O'Donnell, who about that, and the the gap sizes on average are about 2 to 500 base pairs. So they're not that big. And it's when they get to be kilobases that is when you inactivate the synthesis, right? So I think there is some sort of, I'd have to draw some sort of little loop to justify it. That's the bit I don't have. But there's a size-dependent TLS recruitment.
Great. Well, let's thank Simon one more time.